Now we're going to go ahead and start talking about a specific type of operon, the lac operon. And this is going to be a type of negative inducible operon. So on the lac operon, we have a couple different parts that are called I, P, Z together, I, P, O, Z, and Y. So let's talk about what each of these mean. I is talking about your repressor protein. And since the lac operon is a negative inducible, that means that the repressor protein is made active. And as long as there are no mutations, we're going to talk about mutations in a little bit. P stands for the promoter. That's where RNA polymerase will bind to do transcription. O is your operator. That's where the repressor protein will bind to inhibit transcription. Z stands for beta-galactosidase. And beta-galactosidase has the job of breaking down lactose into galactose and glucose. So that's why this is called the lac operon, because its purpose, or the proteins that are made, beta-galactosidase and permease, function in breaking down lactose. So beta-galactosidase does the actual job of breaking it apart, and permease is going to do the job of bringing it into the cell. So permease is going to go out a protein channel onto the cell membrane, so any lactose on outside can go through that protein channel and come inside, where now that lactose is split up by beta-galactosidase into galactose and glucose. We also have a molecule called allolactose, and this is something else that beta-galactosidase can do. It can either directly break up lactose into galactose and glucose, or it can change it a little bit and turn it into allolactose and then break it up. So allolactose is very, very similar to lactose. And allolactose is actually going to be our inducer molecule. So remember, in negative inducible operons, we have an inducer molecule that is going to go turn on transcription. So allolactose is going to be our inducer, and it's going to say, hey, lac operon, lactose is here. You need to start transcribing because we need to get rid of this lactose. We need to break it down. So since it's an inducer, that means it's going to bind to the repressor protein to make it inactive. That way RNA polymerase can now go bind to the promoter and start transcription. So let's talk about some of the notations in IPOSI. We're going to do a couple here and a couple on the next slide. Starting with I, there are 3 different ways you could see this. I plus, I minus, IS. I plus means everything is totally normal. It's exactly what we want it to be. The repressor protein is made in active form. It binds to the operator, unless that inducer molecule is there. If the inducer molecule is there, it makes it inactive. So everything functions just like we would expect in a negative inducible operon. I minus means that the repressor is going to be made in a mutated form that cannot bind to the operator, meaning it can't repress transcription. And finally, IS, this is going to be a super repressor protein. And this means that it's going to go and bind to the operator, just like I plus does. But with I plus, if that inducer comes in and say, hey, repressor protein, get off, the repressor protein will be like, all right, I got you, I'll get off. Super repressor protein looks at the inducer molecule and says, I don't care what you say, I'm not getting off. So the super repressor protein is always bound to the operator, even if the inducer molecule is there. The inducer molecule cannot make it leave the operator. It can't make it inactive. So that means that our regulatory protein is always active. It's always bound to the operator, and transcription is always repressed. The next one in IPOSI is P. You can have P plus or P minus. P plus means our promoter is working. RNA polymerase can bind just like it's supposed to. P minus means that your promoter is mutated and RNA polymerase cannot bind to it. If RNA polymerase can't bind, that means that it can't move down to make mRNA. So if our RNA polymerase can't make mRNA, the genes aren't being transcribed at all. All right, now let's look at O. So for O, there are 2 types. You can have O plus and OC. And OC is like the equivalent of having a minus. We just use a C there to say constitutive. So, or constitutive, O plus, the operator is working and the repressor protein can bind. So everything is normal. OC means that it's mutated and the repressor protein cannot bind. If it can't bind, that means that there's nothing to stop RNA polymerase from binding to the promoter. So the transcription of genes cannot be inhibited, and that means that the genes are always being transcribed. Your Z is your beta-galactosidase, Z plus and Z minus. Z plus means it's made in an active form. Z minus means it's made in a mutant form. Same idea for Y, which stands for permease. Y plus, permease is made in an active form. Y minus, permease is made in a mutant form. So now let's talk about how we talk about mutations. Whenever we're discussing mutations, we're always going to be talking about in the presence or absence of allolactose or lactose. Oops. So there's 4 categories that you'll see. Beta-galactosidase in the absence, permease in the absence, beta-galactosidase in the presence, permease in the presence. So let's look at a completely normal lac operon, plus is everywhere, everything is functioning how it's supposed to. What happens in the absence and the presence of lactose? Well, in the absence of lactose, that means there's no lactose, meaning that there's no allolactose. If there's no allolactose, there's no inducer. That means our repressor protein is going to be bound to the operator, stopping transcription. RNA polymerase can't bind. If there's no transcription, that means we're not making beta-galactosidase, we're not making permease. So in the absence of lactose, nothing's being made. In the presence of lactose, we would have lactose and we would have allolactose. Allolactose would go bind to the repressor protein, making it inactive. The repressor protein is no longer at the operator, meaning that now RNA polymerase can bind to the promoter and begin transcription. So absence, no beta-galactosidase, no permease. Presence, we do have beta-galactosidase and permease. So here are some steps for solving mutations if you're only given one strand. We're also going to talk in a little bit about partial diploids, where you would have 2 strands of your lac operon and you have to kind of look between them and see what's happening. So step 1, you're going to check for a super repressor. If you have a super repressor and the operator is not mutated, you can just automatically cross off Z and Y. Because if you have a super repressor and it can bind to the operator, it's never leaving. If you don't have one, or if your operator is mutated, we'll go to step 2. Step 2, check for a promoter mutation. If P is mutated with P minus, cross off Z, cross off Y. If you don't have that mutation, you would go to step 3. Step 3, we're going to look at just C and Y, and we're going to see if either of them is a minus. If either of them is mutated, cross it off, and then go to the next step. And if at any point you cross off both Z and Y in the strand, that means that that strand is not going to produce Z or Y no matter what. Step 4, we're going to look for a mutant repressor. We're looking for I minus. If you have I minus, at this point, anything that's not crossed off will always be produced, because this means that we're never able to repress transcription. So anything that we can make, we will make. And if we have I plus instead, we would go on to step 5. And step 5, we're going to look at an operator mutation. So at this point, we know we have I plus. If O is mutated, that means that, again, just like with I minus, even though we're making the repressor molecule, it can't bind. So anything that is not crossed off at this point will always be produced because there's nothing to repress transcription. But if O is not mutated, so it's normal, and we have the I plus, that means that everything that is not crossed off will only be produced in the presence. Because in the absence of lactose, in the absence of our inducer molecule, that regulatory protein is going to be bound to the operator and repressing transcription. In the presence of lactose, we have that inducer molecule, regulatory protein leaves, now we start transcription. So first we're going to do some practice with 1 mutation, and then we're going to do some practice with 2 mutations, and then we'll go to diploids. So the steps that I just gave you are a lot more helpful if you have like 2 or 3 mutations with 1. There's not really a point of going through the steps saying, look for this, look for this, look for this, look for this, because there's only 1 mutation and everything else is normal. So I'm not going to follow the steps here. I'm just going to talk about what would happen in each of these. So if we have I minus, that means that the regulatory protein that is being made is not able to bind to our operator because it's made in some mutant form. If it can't bind to the operator, that means that we cannot stop transcription. That means that RNA polymerase is always going to be transcribing, no matter what. So we're going to be making beta-galactosidase and permease, regardless of what the transcription is, regardless of what the condition is, because RNA polymerase can just always bind to the promoter and keep going. There's nothing to stop it because our regulatory protein is a mutant. In the second one, our promoter is mutated. That means the RNA polymerase can't bind. If RNA polymerase can't bind, we can't make Z and we can't make Y because we're not able to transcribe at all. So no matter what the conditions are, we're never going to make anything. RNA polymerase can't bind. In this third one, we have OC, which is like having a minus symbol, it's just saying it's mutated. So that means that our regulatory protein cannot bind to our operator. If it can't bind to the operator, it's just like that top one where we can't stop transcription from happening. So transcription is just always going to be happening. In our next one, we have Z minus. That means that beta-galactosidase is always going to be made in a mutant form. So automatically, I'm just going to put this minus symbol to show that there's none of it. If it's always made in a mutant form, it doesn't matter if there's lactose or not, always mutant. So now we would look and see if we had anything else that was messed up. Since these are all one mutation, we don't. That means that our regulatory protein is made in an active form. It binds to the operator whenever there is no inducer molecule. So with no lactose, our regulatory protein is bound, not RNA polymerase, meaning we're not transcribing anything. So in the absence of lactose, we're not going to make any permease because there's no transcription happening since our regulatory protein is bound to the operator. However, in the presence of lactose, that allolactose inducer molecule is going to come in, bind to the regulatory protein, make it leave the operator, and now RNA polymerase can come in and start doing transcription, meaning that it will make permease in the presence of lactose. For our next one, this is kind of the same idea. Y minus, meaning we're not going to have permease no matter what, because it's only going to be made in a mutant form. For beta-galactosidase, that is a plus, which you can't tell because I kind of drew over it. So that means that we will be making it. We need to look at the conditions. Same idea as before, since everything else is normal, usually without our inducer molecule, our regulatory protein will be bound, transcription will be stopped. No beta-galactosidase in the absence of lactose because we don't have that inducer. If we are in the presence of lactose, we have that inducer molecule, meaning that the regulatory protein will leave the operator, RNA polymerase can bind and start transcription. And finally, the last type of mutation we have is IS, which is a super repressor. And assuming that our operator works, which it does here, that super repressor will bind to the operator and never let go. It is always, 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 always going to be there. It doesn't care if there's lactose or if there's not lactose, it is stuck there. Meaning that transcription is always blocked. You're not going to make anything. So now let's go ahead and look at what happens with two mutations. And for two mutations, we are going to look through these steps of look for a super repressor, check the promoter, check for Z and Y, check for a mutant repressor, and then check for operator mutations. So starting at this top one, I'm going to run through those steps. So step one is look for a super repressor. We don't have one. Step two is look for a promoter mutation. We don't have one. Step three is look for a Z or Y mutation, which we do have one. We cannot have Z. Z will always be made in a mutant form because it's a minus. So I'm just automatically going to put a minus for my beta-galactosidase because of that minus symbol on the Z. And then my next step is looking to seeing if I have I minus or I plus. And I have I minus here. So at that point, I know that that super repressor is made in a mutant form. It can never bind to the operator, meaning transcription can never be repressed, meaning transcription is always going to be happening. So permease will be made in the presence and absence of lactose. Looking at this next one, step one, check for a super repressor. Again, we don't have one. Step 2, check for a promoter mutation. We don't have one again. Step 3, check for mutation in Z or Y. Y is mutated, meaning we're never going to have permease. And step 4, check for I plus or I minus. In this case, we have I plus, which means that we go on to step 5, which is looking at your operator. If the operator is mutated, then everything that hasn't been crossed off will always be produced. If the operator is functional, everything that isn't crossed off will only be produced in the presence of lactose. In this case, it is mutated, meaning that even though our repressor molecule is made, it can't bind. That means transcription is, again, never inhibited, and beta-galactosidase will always be made, regardless of if there's lactose or not, because we can't inhibit transcription because that regulatory protein cannot bind to the operator. Okay. So now we've looked at just one single strand, but there's also another way that you could see this, and this is with partial diploids. In bacteria, this would be like having plasmids in the cell, which are just little rings of DNA that could have another lac operon on there. So you could have 2 lac operons. So the super important thing with this, it's going to be basically the same steps. The difference is with your I. So with diploids, I is going to be transacting, meaning that whenever we have our regulatory protein, if it's made on the top strand, so remember, let's say that this is my operon, and right here is where I make my regulatory protein. Just because I make the protein here doesn't mean it has to bind here. Because once it's free, it can bind to this top one, or it could bind to the bottom one, even though it's made from the top. So if you only really need one, and it can affect both strands, because if this top one is functional, it's I plus, the bottom one is I minus, so there's no active protein made from here. This top one can make active proteins. It'll send an active protein to the top, and it sends an active protein to the bottom. So I plus trumps I minus, meaning if you have one I plus and one I minus, there will be a repressor bound to both operators as long as your operator isn't mutated. In addition, IS trumps I plus, or by default I minus, meaning that if you have at least one IS, a super repressor will be bound to both operators, again, as long as the operator is not mutated. So in order to account for this, step 1 is a little bit different than step 1 earlier, in that in order to account for this transacting, I want you to just change it to where both of them say the same exact thing. So for example, if you have an I plus and an I minus, I plus trumps I minus, make them both an I plus. If you have IS and I plus, make them both an IS. Because now we can just look at each strand individually without having to be like, okay, wait, but do I need to look at this top one? No, we don't have to do that. Just change them so they say the same thing, and then you can look at each strand on its own. And at this point in step 1, if you do have an IS, check both strands for functioning operators. If it is functioning, you can cross off Z and Y in that strand, and technically be both strands because you would have 2 ISs, and nothing would be produced. So for step 2, we're going to do the same thing that we did before in step 2, which is look at the promoter mutation. And keep in mind that everything here is always going to say in that strand. Just because I have a promoter mutation on the top does not mean that I have it on the bottom. So once we change all of our I's in step 1 to say the same exact thing, you can just look at one strand at a time. And if you have a promoter mutation, you have P minus, cross off the Z and Y just in that strand. Step 3, check for Z's and Y's for all the ones that you haven't crossed off yet, and cross off any that have a Z minus or a Y minus. And you're always going to be looking in both strands. And at this point, if you had 2 I minuses, whatever is not crossed off will be produced in the presence and absence of lactose because our repressor doesn't work. And for our next step, if you have an I plus, you're going to check your operator in each strand to see what happens. So if we have I plus, but our operator is mutated, that means that our repressor molecule can't bind there. So whatever has not been crossed off will be produced in both presence and absence of lactose. If the operator is not mutated, so it's functional, whatever has not been crossed off at this point will only be produced in the presence of lactose. So let's do an example, and I'm going to rewrite this so that they're on top of each other. So we have I minus P minus O plus Z plus Y minus, and I plus P plus OC CY, or sorry, Z minus Y plus. Step number 1, change my I's to say the same thing. I plus trumps I minus, IS trumps I plus, and I minus. So I have one I plus, the other's an I minus. I plus wins. I change the top one to say I plus. And now I'm just going to look at each strand individually for every single step from here on. So at this point, I would check for a super repressor, which I don't have. So I'm going to go ahead and go on to step 2, which is looking at my promoter. And anytime that I have a mutated promoter, I cross off Z and Y in that strand. So right here, I have a mutated promoter. I cross off Z and Y in that strand. If RNA polymerase can't bind, transcription can never start. So in that top strand, I'm never going to make Z or Y. But that doesn't affect the bottom strand because RNA polymerase can bind to the promoter on the bottom strand. So that's step 2. Step 3 is looking for Z's and Y's and crossing anything off that has a minus. And since I only have 2 options left, only one of them has a minus here. I'm never going to make functional beta-lactosidase because of that minus symbol. And I've already crossed off the one on the top because of that mutated promoter. So now my only option is to produce permease, but we need to see when it's being produced. So now we're going to go look at our I again. And since it is I plus, that means that now we need to go look at our operator. And remember, if the operator is mutated, that means that whatever is not crossed off, which is our permease, our Y, will be produced in both the presence and absence of lactose. If that operator is functional, whatever is not crossed off will only be produced in the presence of lactose. So because this is mutated, that means that permease will be produced in the presence and absence of lactose. All right. So obviously there are a lot of steps here and there's a lot of conceptualizing and thinking behind it. So I'd recommend just doing a lot of practice problems with these, get familiar with the steps, and not only just memorizing the steps in the order that they go in, but really be able to understand why I put them in that order, why certain things happen, because it'll make it a lot easier to remember if you can understand the mechanisms behind it instead of just memorizing the words. So I just really recommend getting a lot of practice with this, because once you have done quite a few, then the process starts to make a little more sense and you start to get really good at it. But your first couple that you do will take you a little bit longer and they will be a little bit more confusing. So just try and get as much practice in as possible. And that is the end of our video on the lac operon.
I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.